Today I'm going to be talking about a 3D chaos game encoding for DNA sequences that facilitates the comparison of those sequences. To provide some background, DNA is a hereditary material that encodes information through 4 chemical bases. The order of those bases provides information about how to build and maintain an organism, and tracking the accumulation of mutations, such as deletions or substitutions, will allow us to follow the evolutionary trajectory of an organism. The 3D chaos game representation starts with the 4 nucleotides assigned to vertices of a tetrahedron. And the encoding of a DNA sequence in this representation starts at the origin and moves iteratively halfway toward the vertices in the order specified by the DNA sequence. An important element of the chaos game representation is that it is in a way self-aligning. Regardless of how sequences start or end, matching subsequences will have points in this representation that begin to converge in space the longer the sequence matches. Now, in order to calculate distances between DNA sequences in this representation, I look to some shape similarity techniques developed for comparing 3D chemical structures. The first method that I use is based on the volume intersection of the 3D representation. I use Gaussian spheres at every point to estimate the total overlap in volume, and that overlap will quantify sequence similarity. I also explore 3 additional methods based on shape signatures. Essentially, histograms are used to represent distributions of structural features in this representation, and those histograms will then re-represent the DNA sequence as a vector of frequency counts. The counts are centered and scaled prior to calculating a Euclidean distance. The features that I explore for this are the edge lengths in a 3-point sliding window of the representation. I refer to it as the edge signature. I also explore the angles in a 3-point sliding window, which I refer to as the angular signature. And then I also built histograms using just the coordinates of this representation. Now, in order to assess the performance of the distances generated, phylogenetic trees were generated and compared to those built using well-established multiple sequence alignment methods. I have here results from analyzing the coding sequences of the first exon of the beta globin gene. And the top are results using the volume intersection method, as well as the angular signature. Other features produced results that were very similar. On the left is a tree generated after aligning with cluster omega. And the primary difference between shape-based methods and the alignment-based methods can be seen in this branch of the tree here, where the chimp, human, and rabbit are. I've highlighted the sequence of those organisms down below. And the sequence alignment shows that there are 9 substitution mutations between the rabbit and the human, and there are 13 additional insertions between the human and the chimp. Essentially, for alignment-based distances that penalize terminal gaps, then the rabbit will always tend to be closer to the human than the chimp is to the human. And that's because the number of mismatching base pairs between the rabbit and the human is smaller than the number between the chimp and the human. There are also alignment-based distances that do not penalize terminal gaps. In those instances, those methods identify the chimp as being identical to the human, despite the obvious difference in their DNA sequence. In contrast, all 3D chaos game representation methods that I have proposed place the chimp closer to the human without asserting that the 2 are identical. The insertion of the 13 additional nucleotides here is likely the result of a single mutation event. So it's probably more accurate that the 3D chaos game representation places the chimp closer to the human than the rabbit to the human. And it does so without falsely identifying the chimp as being identical to the human. Simulations also show that the methods proposed produce distances induced by deletion mutations to be very similar to the distances imposed by substitution mutations, regardless of parameter settings. Additional tests on simulated phylogenies also show that my proposed method is robust at identifying sequence similarity even in the face of recombination events. For example, alignment-based methods will doubly penalize a transposition mutation as both an insertion in one location and a deletion in another. In contrast, these shape-based metrics only penalizes the transposition mutation once. In conclusion, the proposed methods perform competitively with well-established alignment-based methods. These shape-based methods based on signatures and histograms run very quickly, which is highly desirable. There are some parameter selections that must be set prior to analysis. One parameter that must be selected is the size of your Gaussian sphere. And another downside to the volume intersection method is that it is very computationally intensive.